So as we get ready to start talking about eukaryotic cells, somebody remind me of what gene regulation looks like in prokaryotic cells. How do prokaryotes regulate gene expression? Yeah, Alyssa. What's that? Well, they do a little bit. With what? What do they use? The operons. There you go. And there are 3 upstream pieces of any operon, right? So an operon is a suite of genes, right? The lac operon, it's 3 genes. The trip operon is 5 genes, all packaged together. But there are 2 upstream pieces that influence gene expression. And what are they? The promoter. And what does the promoter do? This is true of prokaryotic and eukaryotic cells, by the way. What does the promoter do? Yeah, it recruits RNA polymerase. It tells it where to bind and where to begin transcription. And so the very first part of any transcript is actually a portion of the promoter. Okay? All right, and then what else is there? So there's the promoter telling RNA polymerase where to bind, recruiting RNA polymerase, if you will. And what else is there? Gives the name to the operon. Starts with the same 4 letters. Operator. You've got the operator at the beginning upstream of every operon, and what happens there? RNA polymerase does not bind at the operator. It binds at the promoter. The promoter tends to be upstream of the operator. The repressor binds to the operator and inhibits RNA polymerase from transcribing. Okay? And so you have inducible operons, which the default position is what? Off, and they can be induced and turned on by impacting how the repressor binds. And then you have silenceable, or you have so you've got inducible and repressible. Thank you. There's the other term. Repressible operons, where the default position is what? Is on. So the default position is that your repressor is inactive and the repressor has to be activated. Whereas inducible operons, like the lac operon, the default position is the repressor is active and needs to be deactivated. Okay. Either way, that's the control of gene expression. And that's the sum of it in prokaryotic cells. Prokaryotes tend to have small genomes compared to eukaryotic cells, even compared to single-celled eukaryotic cells, and therefore gene expression is a little bit simpler. All right, so here is the structure of a eukaryotic gene. The first question we're going to deal with is, what is the structure of a eukaryotic gene? We need to review some of this in order to get a better appreciation of what's going on with the regulation of gene expression. So take a look at this. And what is here that you don't find in prokaryotic genomes? What do you see? What's that? An enhancer. And notice this little symbol here. This is telling you that this is really far upstream. Okay? This is showing you that you're skipping a number of bases. And so significantly distant upstream is a region called the enhancer region. And what do enhancers do? What does it mean to enhance something? Make it better, right? You've got the little enhance wand on your phone, right, that you could do, and it enhances your images. Okay, and so what this does is it doesn't impact necessarily how RNA polymerase binds. It impacts how easily RNA polymerase can carry out the transcription. The promoter is still going to recruit RNA polymerase, just like it does in prokaryotic cells. The enhancer increases the likelihood of RNA polymerase getting through its transcript. The quicker it gets through the transcript, the more you can transcribe it. Okay? Anything else in this that is not in prokaryotic genes? So we've got the enhancer that is not in prokaryotic genes. What else? Yeah. So the promoter is a little bit different. You've got the TATA box or the TATA box in eukaryotic genes, and the entire promoter region is significantly larger. Okay? The promoter region is significantly larger there are several different types of RNA polymerases, and each gene is responsible for attracting a certain combination of transcription machinery. And then anything else that you see that's present in eukaryotic genes and not in prokaryotic genes? And it impacts the second part of gene expression. First part is transcription. The third part is translation. The second part is RNA modification. Yeah, Alyssa. Exons and introns. Remember, one of our RNA modification is splicing. And so eukaryotic genes have sections that get cut out of genes. Parts that get transcribed but don't get translated. Okay? Parts that get transcribed but don't get translated. These are all unique features of eukaryotic genes and shape how eukaryotes have to regulate gene expression. So they contain a promoter region upstream from the transcription unit. This is similar to prokaryotic cells. The exact nature of the promoter is different. It's got a TATA box, and if it's a protein-coding gene, it recruits RNA polymerase II. You have more regulatory elements, the promoter proximal elements. These slow or enhance the rate of transcription. And then even further upstream, you've got an enhancer that enhances or slows the rate of transcription. And so you have a lot more pieces that can control the rate of transcription, control the rate at which RNA polymerase functions. When we were talking about transcription before, I used the analogy of a road as RNA polymerase or even DNA polymerase as it's replicating the genome is moving along a smooth path, and what tends to disrupt it is hitting a bump, right? Like if you form that palindromic sequence and it kind of locks everything up, or if it puts the wrong base in, it creates a bump, right? And so if you were going to slow or enhance the rate of transcription, how would you do it? How would you slow the rate of transcription or enhance the rate of transcription? Thinking about that analogy of a road, and RNA polymerase as some kind of heavy machinery moving along this road.
You put a bump in the road.
Okay, and how would you do that? What would that look like in terms of our DNA template? What would a bump look like?
Something that's like twisted.
Okay.
Like a loop or something.
Okay. Just some really complex three-dimensional structure to it. And that's exactly what you see oftentimes with this control of gene expression. It's controlling the shape of the chromosome. Certain chromosomal shapes facilitate faster transcription than others and more transcription than others. Did you have a question, Anna? Okay. Charles?
Why does the rate of transcription matter?
Because the rate of transcription dictates how many transcripts you can make, and then ultimately how many transcripts that can be translated, and how much protein you can make.
Also, it regulates the rate of protein synthesis.
Yes. Even if it's not a protein coding gene, it's still going to influence protein synthesis, because there are other RNAs that aren't translated but regulate translation in some way, either directly or indirectly. All right? So next question we have to deal with. What are different ways in which eukaryotes regulate expression? Regulate expression. Bless you. And so I mentioned to you that prokaryotic cells can only regulate transcription by inhibiting it, right? So you've got an operator that if the repressor binds there, it inhibits transcription. Why is that the only way that a prokaryote can regulate gene expression? Yeah.
So does it regulate it past transcription level?
Why not?
Doesn't have a nucleus, right?
Right, it does not have a nucleus, which means what happens at the same time as transcription? Translation. At the same time that you are transcribing an RNA transcript, it is being translated by ribosomes. Okay? Those happen simultaneously. And there's no RNA modification like there is in eukaryotic cells, so you can't regulate it there. Eukaryotic cells don't have the same limitations. They can regulate the transcription by enhancing or slowing the rate of transcription. They can completely shut it down. We saw that, we've talked about that with X chromosomes, right? Female mammals have typically have two X chromosomes, and one of them needs to be shut down, right? And so you could shut down the entire chromosome by condensing it. And this is what happens during mitosis and meiosis when we can physically see the chromosomes. They are condensed, and it also shuts down transcription, which is good because you don't want that going on when you are trying to separate your chromosomes during mitosis and meiosis. So they can regulate at the transcription level, enhancing, slowing the rate of transcription, entirely shutting it down. They could regulate it during the modification, RNA modification, by determining which introns get cut out, even destroying your pre-mRNA before it matures into mRNA. They can regulate translation by destroying your mature RNA before it leaves the nucleus or even out in the cytosol. And then they can regulate in post-translational as well by destroying proteins. They have eukaryotic cells have really complex machinery for destroying proteins.
Why would you go through making a protein and then just start destroying it?
Because remember, cells typically are making proteins in response to some signal, right? And they'll start making that protein, but then that signal may dissipate and disappear, and they no longer need that protein. And so you don't stick it around. You don't commit to it just because it cost you a lot to make it, right? It's never a good idea of committing to something just because it costs you a lot of money to get it, right? Sometimes it's still wasteful. Anyways, I have a good story about that, but we'll save that for another time. All right. So to sum this up, eukaryotes can make short-term changes by turning off and turning off genes in response to signals, environmental conditions. Multicellular eukaryotes tend to make long-term changes as cells differentiate, right? Nerve cells are going to express different genes than liver cells, which are going to express different genes than skin cells. And so some of those are long-term, relatively permanent changes in gene expression, although can be overwhelmed by exposing those cells to the environment of the embryo. And so you can people have done this kind of work a lot with plants and some animals, where you take a cell and you put it back in the environment that the early embryo experiences, and the cell converts back to what you call a totipotent cell, a cell that can differentiate into anything. Yeah. Yeah, but it's an adult stem cell instead of an embryonic stem cell. So nuclear DNA, we talked about this before. It's organized with histones, and so the DNA is wrapped around these histone proteins, and even histone-like proteins. And so the tighter you wind the DNA around those proteins, the harder it is for that machinery to access that DNA template. Nuclear envelope separates transcription and translation. And so gene expression can be controlled transcriptionally, post-transcriptionally, translationally, and even post-translationally. All right, so these are some of the things that eukaryotic cells can do that prokaryotic cells cannot do. I mean, you might argue that prokaryotic cells don't need to, and eukaryotic cells do, but that doesn't change the fact that these are some things that eukaryotic cells can do and prokaryotic cells cannot. All right, so we're going to take a lecture break right now because it's time. And what I want you to do is this. As you start to think through some of these things and working with those around you, I want you to come up, based on the structure of a eukaryotic gene and how gene expression works in eukaryotic cells, I want you to come up with a list of ways or a list of instances where a eukaryotic cell can control gene expression. Okay? So if you're going to put it into the categories of transcriptionally, what would that look like? Or post-transcriptionally, what would it look like? Translational modification, what would that look like? And post-translational. All right, take a couple of minutes, work with those around you, and come up with a list of ways in which a eukaryotic cell can regulate gene expression.
Hi. Three of us will be a group.
That's all we need.
That's all we need. Okay, so we're going to pull up four ways to do. You have transcriptional. Let's start with each one of those.
Post-transcriptional.
Let's start with transcriptional. So every time I hit into this, how can it be controlled transcription? So like by changing the shape of the chromosome, right?
Yeah. A question that is yeah. To make it easier or harder for the polymerase to.
So changing the shape of the chromosome. Okay, what's the next one?
Post-transcription.
So obviously after it's transcribed, it can't be translated to move down the nucleus, right?
So you have to regulate how it's transportation.
Regulate transportation.
I mean, already all
Slides show you the answers there.
Translational regulation, deregulating ribosomes.
Translational regulation.
So like translational, right? Make
how
translate.
fast.
Is it really complicated?
Yeah, it's how fast or if it does or whatever. Lock it, enhance it, whatever else you do. So yeah, it's transported and
You got a little bit of time to wrap up your thoughts and see if you can find somebody in your group that wants to share your thoughts.
Translational is basically ribosomal enzymes.
All right. What are some of the ways? Do we have any groups that want to talk about how a eukaryotic cell can regulate gene expression at the level of transcription? Alyssa.
Oh, then, well, we talked about the bumps in the road, so like
Uh-huh.
complicated things sticking out, and then kind of makes it go slower.
Sure.
And then the other way is if there's no enhancer or an enhancer that can dehancer and makes it slow.
Sure.
And then, and I guess repressors.
You do have repressors. They don't bind to an operator just upstream of the gene, like in prokaryotic cells, but there are repressors in eukaryotic cells. Mm-hmm. Yeah. Any other thoughts? All right, any groups want to share some thoughts on how a eukaryotic cell can regulate gene expression at the post-transcriptional level, at the level of RNA modification? Yeah, Daniel.
I was going to say, like, during meiosis,
Okay.
all the different checkpoints that the cell is giving off proteins, apoptosis to take in, destroy that cell.
Sure. I mean, that's an irreversible change, but it is a change nonetheless of controlling levels of gene expression. Any other thoughts?
We were just saying like it can regulate its transportation out of the nucleus.
Sure.
It's in the nucleus, it can't be translated until it's moved out of the nucleus.
Right.
So
Yep. And it's got to be labeled in a particular way. The poly-A tail tends to provide the label necessary for it to be transported out of the nucleus. Yeah. Yeah, Alyssa.
I have a question.
Sure.
If there's like too much of a protein, like something went wrong and there's like way too much of it, and they're like, oh snap, we gotta get rid of all of them. Can they mark it like they would of something they don't want?
Yes.
And, like eat it?
Yes. Mm-hmm. Yeah, we'll talk about that. It's post-translational modification. And so, yeah, you can tag proteins for destruction. Yeah. Yep, and then you eat them. Mm-hmm. Yeah. It's like walking through with sticky notes and labeling garbage and keep, right? Keep, toss, keep, toss. None of you do that? Like at the end of a school year, you label things so that your roommate doesn't throw them away? I guess you could just not bring very much stuff or just throw everything away at the end of the year, right? And start anew. I don't want to keep any of this. My roommate touched it and it's gross. No? None of you have gross roommates? Oh gosh. I wish I could have made it all the way through college without gross roommates. All right, any other thoughts? So remember with splicing, the cells cut introns out of your pre-mRNA. There are different ways in which those introns can be cut out. You call that alternative splicing, and it produces different protein products. Okay. What about any groups want to share how it can control gene expression at the level of translation? So now the mRNA, it's mature. It left the nucleus, right? It's a eukaryotic cell. Translation does not take place inside of the nucleus. Translation takes place either in the cytosol or attached to what? Where else do you find ribosomes? So it always is on a ribosome. It's either on a free ribosome or it's on a ribosome attached to what? The rough endoplasmic reticulum. Either way, translation is taking place on a ribosome. So how do you control gene expression at that level? Because once translation starts, it's going to finish. Yeah. So those would be post-translational, but you absolutely, you can modify how the protein is folded, which is going to change the protein. Yeah.
The rate at which proteins are synthesized?
Sure. So how would you change that? How would you change the rate at which the proteins are translated? Remember, your mRNA is already mature. It's left the nucleus. So you either need to tag it for destruction so that it doesn't get translated, or you could chemically modify it so it loses the 5' untranslated region. Remember we talked about that, the upstream that signals it where to bind to the ribosome. Or you have RNAs that are made of nucleic acids, and nucleic acids like to make hydrogen bonds with other nucleic acids, right? So you have RNAs that are called interfering RNAs. We'll talk about this a little bit, that can actually bind to that mRNA and prevent it from being translated. Because if there's something bound to it, you can't read the codons, right? The anticodons can't come in and bring their particular amino acid. And then post-translational, you can change the way the protein is folded, or you can mark it for destruction. So here are the transcriptional modifications. Here's showing you a series of transcription factors that bind to the promoter and bind to the proximal promoter complex, all to ensure that RNA polymerase binds to the correct place. Do you remember when we talked about the lac operon? That it wasn't just the it wasn't just the presence of lactose that turned it on. You also needed something to be absent. What was that? Glucose. Because when glucose was absent, it activated the cap protein that helped RNA polymerase bind. And so in eukaryotic cells, RNA polymerase recognizes the promoter, but it can't bind without these initiation factors, these transcription factors. And so there are parts in this promoter region that help RNA polymerase bind to the correct place. If they don't help RNA polymerase bind, it will bind, but it will take a really long time for it to find it. It's like without an enzyme, that reaction will probably still happen, but it will take a long time for that reaction to happen. And so here are some of the shapes of the proteins that you find that act as transcription factors. Here's a protein with a helix, a turn, and a helix. It actually sits in there and helps to unzip and create some instability in the two strands of protein and help RNA polymerase to bind. Another one, the zinc finger, the leucine zipper. And then here you have the enhancer noted here that's actually helping to build this complex machinery, ensuring that RNA polymerase gets there as quickly and as efficiently as possible. All right. So sum this up. Transcription factors have to bind to the promoter in the area of the Tata box and in the area of and form what you call the initiation complex. Then there are activators that bind to the proximal elements. There are activators that bind to the enhancers and that help to get RNA polymerase in the correct place and help it to move as quickly and efficiently as possible as it works through transcription. The faster transcription happens, the more product you make, and the better the cell responds to some particular some signal. Now, there are repressors in eukaryotic cells just like there are in prokaryotic cells. And so this next question deals with all of the post-transcriptional modifications of gene expression. And I'm going to focus just on the regulation of translation. Now, all of these are going to tend to be in response to some kind of a signal, some kind of a stimulus. And so here you've got some signal molecule, some ligand bound by a receptor. In this case, the receptor is inside of the plasma membrane. What does that tell you about this ligand? If the ligand binds to a receptor inside of the plasma membrane, what does that mean about the ligand?
Probably water soluble and small.
Well, if it were water soluble, it'd have a hard time moving through the plasma membrane. Small, you're onto the right track. It's probably nonpolar and not water soluble, right? If it can move through these big regions of hydrophobicity inside of the plasma membrane. So most of those are really small steroids. Steroids are lipids, and if they're small enough, they can diffuse through the plasma membrane, and so the receptor can oftentimes be on the nucleus. And so it's still, we've got the 3 steps of signal of the response to a signal. And what are those 3 steps? Step 1, you bind the signal. Step 2, you translate the signal. And step 3, you respond to the signal. And so here it bound? I don't even know how you would make that past tense. The receptor is bound to the ligand, then it is translated into something that moves inside of the nucleus, and then the cell responds to that by regulating the level of transcription. And I told you I was going to focus on regulation at the level of translation, but here just in terms of talking about somewhat permanently changing levels of gene expressions. I'm going to do this and move ahead to the level of translation. All right. So the question we have here is what changes can be made during RNA processing? And then this bleeds over into how we regulate it at the level of translation. And so again, these mRNAs, they've now left the nucleus, they are mature, and they have to bind to a ribosome. And then you have to read the codons 3 bases at a time and translate them into a sequence of amino acids. Yes? But it's a string of nucleotides, and a string of nucleotides can form hydrogen bonds with another string of nucleotides. And so here you have microRNAs, or you call them interference or interfering RNAs, and they can go in and physically bind to your mRNA and prevent the translation. And so these are RNAs that the cell can transcribe and release into the cytosol, into the cytoplasm, to inhibit the translation of all mRNAs or the translation of specific mRNAs. All right, so what I want you to do is take about 90 seconds to 2 minutes. I want you to talk with those around you, and I want you to come up with how would you prevent a specific RNA from being translated, and how would you prevent any RNA from being translated? All right, so if you were a cell and you were going to regulate gene expression at the level of translation, how would you target a specific RNA, and how would you target all RNAs? All right, take 90 seconds to 2 minutes, come up with a list of features, and then we'll be pretty close to done with our class today. All right, take a couple of minutes, starting now.
So for a specific RNA, you need a mini specific RNA, an interference RNA that would bind to a certain codon, on your primitive, and it would be specific to that gene, and that would
start codon. You virtually shut off all translation because they all have the same start codon. Yeah, so I'm pretty sure every gene, like every gene has a start codon that's like the same. It's because that's what tells the ribosome always looks for that start. Right? So it just has to find that start codon and knows to start. So if you absolutely bind to the start codon, it'll shut down all translators. Whereas if you wanted to bind just for a specific one, yeah, you have to go for yeah, not it wouldn't have to be maybe like another you have to make codon that's specific to that. Yeah. I wonder if it's possible, but you have to be careful because they share a lot of codons. So like one codon is codon ABC, the other one is codon CD. If you choose C, you may have shut down this one, but you also shut down that one.
That would be for the only thing is how do you make that? How do you make a microRNA specific to a certain gene like that?
All right, so last time, being Friday, we talked about a couple of questions that I said were really good questions to cover this material from chapter 16. This right here, what changes can be made during RNA processing, this is another really good question that might be asked on chapter 16. Just think about that as you think through this. All right, so what are some ideas? First, let's start with how do you target a specific mRNA? How would you target a specific mRNA? Yeah.
Does it have something to do with like the sequence, the beginning?
It does.
Whatever? So like you have to match something to that specific.
You do. Yeah, so when you are transcribing from the DNA, your strand of DNA is your template that you're writing your RNA from, right? So let's say the piece of DNA looks like this. Okay? And so this is the sequence of the DNA that you want an RNA version of, right? You don't read this to write this. What do you read? You read the complement, right? So you actually read this. So you would read this and then write an RNA version of this, right? And that's how you generate your pre-mRNA, which then can mature and leave the nucleus. If you wanted to inhibit the translation of this, what do you then write? You now write this, right? So if you wanted to, you've already made this, and it's already left the nucleus, but you want to stop translating it. So now you write this, but an RNA version of this. Because then that's going to go out into the cytosol, and it's going to form hydrogen bonds with this that you've already written and inhibit the ability to translate it, right? And that's how you can target a specific mRNA. RNA polymerase II writes mRNA. Which RNA polymerase do you think writes these interfering RNAs? No, it's not It's not a type of tRNA. It's still RNA polymerase II. The same one that wrote this, and then it'll just go and write the complement that will go out and will interfere with it and prevent its translation. What about if you wanted to inhibit the translation of all RNAs for a time? Yeah, Joey.
You could just target the start codon?
Right, every mRNA, every mature mRNA, the translation starts with the same three bases, which is AUG. And so if you wrote an interfering RNA that will bind at wherever it finds an AUG, you're going to be able to inhibit the translation of every single mRNA for a time. But that's dangerous because you don't want it to stick around forever. Now, we talked about RNAs and why it's nice to have RNA be what leaves the nucleus to get translated. Do you remember that? What's true of RNA that isn't true of DNA, besides that it tends to be single-stranded, although it doesn't have to be. It will form base pairs just like DNA will. What's that? It does contain uracil. Yeah.
DNA is a lot more stable.
DNA is way more stable. And so RNAs are very unstable. And there are things in the nucleus that eat nucleic acids. Because you eat nucleic acids in your diet, and you get intracellular digestion that will break up those nucleic acids into components that you can use. And so if you wait a time, you shut down all translation, and wait a time, those nucleases that eat nucleic acids will chew up all the RNA in the cytosol, and you can start over and start fresh after you inhibited all translation. Or you could just inhibit the translation of a very specific mRNA by writing an RNA version of the complement, and you wait enough time, those nucleases will chew up the mRNA, and they will chew up all those interfering RNA, and you can start over. All right? This is what cells do all the time. Yeah, Josh?
Could you just do that by not placing the poly-A tail on?
You could, yep, and that would be modifying it at the level of post-RNA modification or post-transcriptional modification. And so then, absolutely, you could just not put the poly-A tail, you could not put the guanine cap, you could splice it incorrectly, you could just destroy it inside of the nucleus. But if you've already sent it out of the nucleus and it's already mature, but you want to stop translating it, you're left with all you can do is inhibit the translation, right? And wait for it to get destroyed. Alyssa?
Oh, so you're talking about translation, not transcription.
Correct. Yep, regulating at the level of translation.
Oh, because transcription, you just like condense it and it can get.
Right. Yep. Yeah, absolutely. All right. Any other thoughts, questions, concerns? We are currently on slide 35/36 because we'll sum this up. And so we will finish chapter 16 at the very beginning of our time on Wednesday. And then the rest of our time on Wednesday and Friday, we will spend working through group discussions that our quizzes will consist of these group discussions that will go through the study guide that I will post later today on Canvas. All right? Have a wonderful day. Enjoy the rest of your Monday.